It's my pleasure to introduce our next guest from Wave Life Sciences, President and Chief Executive Officer, Dr. Paul Bolno, and Senior Vice President, CMC, Dr. Sri Vadavoina. As a reminder to attendees, if you'd like to ask a question of our speakers, please type it into the question box under the video player. Paul and Sri, thank you both so much for joining us today. I think to start, Paul, could you please provide a couple of minutes of introductory comments about Wave for investors new to the story?
Yeah, no, thank you, Kay, and we appreciate Chardan for hosting us on today's summit, and it's a great topic talking about manufacturing. When we introduce the company and for the subsequent part of the discussion, just a reminder that we're making forward-looking statements during discussion, and so I please refer everybody to our SEC filings for updates. But Kay, as you said, beyond manufacturing and stepping back, since our foundation at Wave, we've really focused on embracing stereochemistry to rationally design therapeutic oligonucleotides. And we believed by doing that, we could drive improved potency, durability, and hopefully outcome for patients. We've actually seen more recent examples of this. So referring to our data update that we provided several weeks ago on our C9orf72 program, where preclinically we had shown data that after 2 doses in a transgenic mouse model, we had seen that translation of potent, durable out to 6-month knockdown of the poly-GP dipeptide, and this is a biomarker that one can measure the impact of taking down the toxic RNA. The clinical update we provided showed that these data that were preclinical were now translating into the human clinical data sets, where we saw at very low doses knockdown of the poly-GP dipeptide, and we'll be continuing to follow these patients out both in the multi-dose setting as well as extended duration. So we'll be extending that follow-up period to 6 months to look at continued knockdown. What's important is not only were these the first data looking at the impact of target engagement with new chemistry and stereochemistry, but importantly, we're excited about the potential for that translation to the other programs in our pipeline. So that's referring to our SNP3 program in HD, where we anticipate data from that study in 2022, as well as in our N531 study, which is our DMD exon 53 study, where we're looking not in CNS and intrathecal, but we're looking at muscle and systemic delivery of our PN chemistry. So great examples of us being able now to see that translation and be able to look at the impact of chemistry across multiple modalities and tissues. And lastly, in taking a step back in that chemistry, really in the ability for us now to unlock in the RNA editing space our AIMer guide strands. And so with that being in the translation to candidate for our alpha-1 antitrypsin program, and we're excited for that to enter IND studies in the 3rd quarter and its progress to the clinic. So really holistically stepping back and looking at multiple, again, modalities, multiple tissues, and leveraging our insights into oligonucleotide chemistry. Stepping back from all of that was an investment that we made in manufacturing and a realization that in the oligonucleotide space, there was a rising tide, no pun intended, in oligonucleotides. And when we think about that, what we saw and envisioned when we were back in 2014 and 2015 was that with more modalities, more therapeutics in the RNA space moving forward, that there would be challenges in terms of meeting bandwidth to deliver molecules for clinical translation. So we made a decisive decision to build GMP manufacturing then to support the growth of our pipeline, to be able to sustain that to the therapeutics that we felt were important and meaningful to advance the patient. When we think about that investment back then, I think we hadn't anticipated in terms of where we are today that the increased demand hadn't been met. So if we think about this kind of constant improvement of more therapeutics, one anticipated, like we were seeing in the gene therapy space, growth of the CDMOs and manufacturers to support those modalities. And what we didn't see was that rising manufacturing capability to meet those demands. So where we find ourselves today is, we have a GMP manufacturing facility. We have a world-class oligonucleotide process development team that's helped us scale and grow our therapeutics into the clinic, while disappointing and remembering the suvodirsen, which was our exon 51 program. We had done a lot in terms of validation of manufacturing processes, synthesis, and capacity to meet large demands for oligonucleotides. So recognizing that we have that capability, we are now focused on utilizing this facility to serve not only Wave's pipeline, but really our underlying promise, which was was to deliver genetic medicine to patients, whether or not we're discovering and developing them, or whether or not we're supporting others in their development of programs. And so we've decided to continue to explore that capability of unlocking that capacity and capability in 2022, delivering therapeutics for patients. So we're excited, to participate in this conference, both as a therapeutic drug developer, but also recognizing that we have a substantial, unique capability in the oligonucleotide field.
Well, great. So with that introduction, can you tell us, what capacity you have online at your facility in Lexington, Mass?
Yeah, so we built Lexington on the manufacturing side to meet demands of really what we saw as where the core bottleneck was. And it's in that translational piece of preclinical into phase 1/2 material. So really, where we think about all meaningful new medicine testing themselves, which is, can you generate material to get through your IND-enabling studies and then ultimately run your human proof of concept study. As we think about the future of medicine, there's always about building capacity for scale-up and commercialization. But where the real unique necessity was is in this phase 1/2 supply. So within Lexington, that is what we have now, which is the capacity to serve both Wave and others phase 1/2 material. We can run multiple runs to increase the amount of material we need. And again, how we've supported a number of our programs. But we have ample capacity beyond ours to continue to unlock that capability within the facility. Shri, you want to add to that?
So I think absolutely agree with you, Paul. So we have capacity to accommodate various programs within the Lexington facility.
I think stepping back too, when we talk about programs, it's easy when we talk about, what Wave's been working on. We've got antisense oligonucleotides. We have exon skipping, splice correcting oligonucleotides. We're bringing a whole new technology forward with our AIMers in terms of RNA editing. And so when we think about that capability, the facility we have, our process development expertise crosses and spans multiple modalities. That includes, not just the uniqueness of our chemistry applied to our oligo, but the ability to serve more broadly others as it relates to long-mer oligos for whether it's DNA editing, CRISPR editing, and the like. Those all require oligonucleotide synthesis capabilities. So kind of getting back again, when we talk about where the need is in the space for oligos, we oftentimes talk about modalities like CRISPR editing or DNA base editing as the biologics, the other side of the house, right? Like in terms of how you deliver the enzyme. Oftentimes forgetting that the guide strand component, that which is capable and necessity to work in conjunction with the enzyme, is an oligonucleotide and requires real process development scalability. And so within our facility, we have the know-how, capability, and manufacturing expertise to lever that.
Yeah, so let's dive into that a little deeper in terms of your capabilities. We followed the company for a while. We've seen the development and maturity of the PRISM design platform. And, initially you focused on synthesizing precisely controlled chirality with your stereopure at each backbone linkage. And, more recently you introduced the phosphoramidate diester modifications or your PN modifications to the backbone. So I think one could argue that these are more complex oligonucleotides to manufacture. So what kinds of specific challenges with respect to manufacturing did Wave have to address to bring these compounds, your own proprietary compounds, into the clinic?
So we've spent a lot of time organizationally and, kind of stepping back and, we're excited to have Shri joining Wave. Now it's been, well over a year, which is exciting as we grow, but kind of stepping back to the original building blocks of Wave, we took on head on, how do we rationally design single drugs? It was a reminder to people that the reason this field had kind of not pushed forward in that space, right? The likes of other stereorandom molecules kind of ignored that space was because the thesis was that you had to try to separate out, you had to pull out or extract some component of a mixture. And what we taught people head on and what we developed internally is not that. That's why we're able to get to yields and scales that are commercially no different than a stereorandom molecule, is that embedded in our chemistry and capability is the synthesis, the design of individual building blocks that control the chirality at each base as it's added. And I think that's important for those who are joining who are not as familiar with oligonucleotide synthesis. You have to think of it as kind of the building blocks for synthesis. What we're doing is we're adding on individual bases with controlled positions. So in a way, if one were, and I know at some point they're going to offer visits on site, it's really fascinating for those who aren't familiar to kind of see the synthesizers in action, because essentially what you're building on a column is the oligonucleotide in its full length. And what we're able to do on each base is control its position through the reagents, design them to link onto the column, and then we shed them off the column. So the scalability is there because you're not trying to extract a percentage of a mixture, you're actually embedding and building the molecule base by base. So that know-how, that capability of doing that for Wave with our proprietary chemistry, but just as easily without the proprietary chemistry in other areas, is really a feature that's scalable and portable across a multitude of modalities. I do think what's interesting, and we'll talk about this, I'm sure, later, is that in building that capability, and not just building the manufacturing capability, or the process development that goes into building the oligos onto the column, there's also the characterization that comes with that. And so the ability for Wave, as we think about now defining the oligonucleotide class, one remembers small molecules, when they have a chiral center, you can have a left hand and a right hand. If you think about thalidomide, it's a perfect example of one that's toxic and one that isn't, right? One position that is toxic and one that isn't. That actually required a whole CMC regulatory body to being able to define each of the parts and then to characterize those parts in terms of what the outcome is. That's really difficult to do in the oligonucleotide space, where you have potentially in a 20-mer 524,288 different substrates. There is a lot of complexity with this from pharmacology and toxicology. So we're also interested in building foundationally in oligonucleotides for manufacturing, is really establishing the process that could be the regulatory CMC process in the future if we establish and characterize individual oligonucleotides as stereopure, as single molecules. And so there's both building the manufacturing processes and opportunities around that, in addition to what we can do beyond that.
Yeah, no, I think that's an important point and does lead me to the next question. And it's been a key topic throughout the conference, and that's on your assays and your analytics. So, in your case, if your desired end product was all right-handed isomers, you certainly had to develop the assays in order to qualify that and make those assessments. So specifically, can you talk about some of the assay development you have in-house and you've developed to assess potency, purity, and stability of your drugs?
Absolutely, sure. Yeah.
So let me start. I think like what Paul mentioned earlier is the difference here is the know-how of finding solutions to either manufacturing or QC testing. And oligos are different from biologics, especially gene and cell therapies. And there are a lot of precedents of RNA therapeutics in the oligo industry. And there are several standardized test methods, primarily focused on identity, strength, and purity. The greatest advantage, what I see at Wave, is there's tons of experience that the scientists develop these methods routinely for all our programs. And we have successfully demonstrated this for more than 6 clinical studies so far, and there are several in pipeline going on. But for each oligo, the standardized methods are modified to accommodate the oligo. So they're different, but it starts with the standardized test methodology.
It's interesting to follow up on that, and I think to Sri's point, I mean, there is a nice feature on the oligonucleotides. We think about them again as long, rather than calling them big, I think we'll call them long, small molecules. But the idea of having very standardized processes, established CMC regulatory processes for validating those packages as they go forward. But to your earlier point, I mean, I think it's also interesting when we talked about kind of the history. When we were looking at how do you do QC, yes, there are established processes for stereorandom drugs, but I think the reason we had spent some time early on manufacturing, really seeing the breakthrough for the industry being chirally controlled medicine, is that when we were looking at kind of the first commercial product in the oligonucleotide field, mipomersen, we had kind of shared kind of the HPLC spec for that. So when you looked at it and you said, okay, what is it? And you see this very large hump, right? And there's just a bunch of different isomers, and it establishes this, wide dynamic range. What's interesting is while you can establish that principle, to Shree's point, you can establish a certain characterization bound. What we were interested in is kind of more the isomeric distribution in that. And I think what was really interesting is that distribution could vary from run to run. So as we think about kind of the diversity of different molecules that are in each batch of a CMC run, it really kind of taught us a lot in terms of our thinking about, how do we create the discipline around having a single spike, right? A single molecule that you can characterize preclinically, characterize it clinically. And so that same batch that you're running safety studies on becomes the same product that you'll ultimately take into patients and potentially commercialize. And so within Wave, building the complexity, not just, on the synthesis, but really building and establishing first principles of how do you create that standardization from a regulatory CMC perspective was one of the core sets of activities within our team in establishing release specs for our individual medicines. So as we think about, building a pipeline of chirally controlled medicines, we've done a lot in terms of manufacturing QC and building new assays. So it's great that, again, we focus on that from our internal manufacturing capability. Those processes, those same controls and individuals, right, we're now able to think about, how do we do that more broadly? So interesting. And I think this is one field that, I keep saying watch the space, because I think as we move medicines forward here, we are planning to really try to reestablish a regulatory standard for CMC for chirally controlled medicines. I mean, that is one of the promises of bringing stereo-controlled drugs forward.
And then you touched on this earlier, but tell us how everything you've learned to date, evolving PRISM, is helping you with the design of the guide RNAs used in your ADAR or AIMer products. Typically we're thinking of a guide RNA with a hairpin to attract the endogenous ADAR, but also if you're going to develop a guide RNA for CRISPR, that might look a little different. But talk about how what you've learned up to this point is going to help you and translate over to your ability to manufacture guide RNAs.
So, I'll focus on us. And when I say us, I'm talking about Wave Therapeutics, right? Our ability to develop and design therapies and treatments. And so, one of the benefits of our AIMer designs are that they're short, chemically modified oligos that don't necessarily require those long structural components that you're referring to. We see that as an advantage because they're short, they distribute into cells more efficiently, they get taken up more effectively. The phosphoguanidine and PN chemical modifications, their positions help get them trafficked appropriately, the right compartment of the cell. And we see that translating, obviously, like we showed in our alpha-1 antitrypsin work, to consequential editing efficiencies of 60%. We're seeing well above 11 micromolars of protein. So we see the therapeutic translation of our AIMer design. We got there because, by controlling backbone and doing rational design, we could look at the interaction between RNA and protein. And that's really the fundamental feature of what we mean by rationally designed oligonucleotides, is by controlling SAR and controlling the interaction between the RNA transcript and the protein that it's going to interact with. We've then taken that and brought that experience onto the manufacturing side, where we're saying, great, if not only could we do that at the scale to develop our SERPINA1 model work that we used in rodent models, but most importantly are scaling that into, getting ready for GMP runs as we think about our IND-enabling studies and ultimately translating that program to the clinic. So we've seen that translation now as we think about getting our processes established for GMP synthesis across, as Shree said, 6 clinical programs to date, and now moving into a whole new modality of editing. So we've got a history of being able to take innovative ideas and translate those into GMP products. I think what's different, and what you're alluding to, is, well, how do we do that more broadly at scale? And I think that's really where our team has the expertise to say, well, we could do it after having controlled stereochemistry and all the features that one needs to scale and do that efficiently. Being able to do that in a longmer design, where we just take an idea where a company might have a really interesting longmer guide strand and they're trying to solve that problem. Well, how do we take this longmer and synthesize it and process develop it and create a meaningful therapeutic for patients? we've got the know-how, capability, and team that's already spent time thinking about how to improve those processes, how to accelerate that, and how to bring a therapeutic forward there. So some of that knowledge is obviously transferable. When I say, the team, I think what's great is we've been able to not just recruit what we would say is world-class oligonucleotide process development folks to Wave to work on our interesting programs, but the retention is the idea of having them work on many problems, right? So how to solve and create the solutions for a multitude of modalities. And I think as an industry, in this environment, it's really important for us to holistically be thinking about where is consolidated expertise, and then rather than dissipating that into a multitude of things, how do we leverage that? How do we take that experience, that network, and be able to support multiple therapeutics, whether or not Wave owns them or others, how to continue to support that ecosystem? And I think that's really been where we've doubled down now to say, how do we leverage that team and let them work on solving really challenging problems across a whole bunch of new areas of oligonucleotide therapeutics.
Okay, great. So Paul, earlier this year, the company did disclose that it would seek to make its excess manufacturing capacity available to third parties. So can you describe some of the parameters around this initiative?
Yeah, I mean, as we, I think it's expanding capacity and really thinking about as we've advanced it, how do we, to that point I was making earlier, how do we lever that experience to accelerate a multitude of therapies? I think the parameters we put around it is, and to our shareholders, I'd say, it won't impact our pipeline. So we've been very deliberate that we built this capacity to support our therapeutic pipeline. Anything we do in this space, we'll have our, we have our capacity locked in. It doesn't impact our therapeutics, but we do have substantially more capacity. We can run a facility if we were running it as a full manufacturing capability to run that 24 hours a day, 7 days a week. That's more capacity than we would be using. So there's an ability to really, within the same footprint, be able to increase throughput through it. That could, continue to double the capacity that we already have in excess. The second piece that's also important is we have scalability of suite. So we purposefully built Lexington with the ability to expand. So within that footprint, we also have the ability to then double that capacity again within our existing facility. And we did that in a forward-looking way, realizing that, if we were going to go through the investment of building that facility, let's make sure it was scalable. And so we have done a lot of things that were very purpose-built to be able to expand that manufacturing footprint in Lexington and then continue to do that beyond. I think our conversations have been extraordinarily productive starting last year into this year with a multitude of companies who, we don't see as competitors. I think we see all of us as working together in a field to advance really challenging medicines to patients who need them. And, we're prepared to work with people to solve complicated problems to bring those medicines forward. And so I think we've purposely built the organization to be able to do that. I'm sure if you have anything you want to add.
No, I think you covered pretty much everything here.
So Paul, with the current stock market valuations, since the beginning of the year, we've seen a number of pre-commercial stage biotech companies announce strategic initiatives designed to extend their current cash runways. And these include, unfortunately, headcount reductions and pipeline rationalizations. But as these companies look to conserve cash, you're likely to see less invest in their own manufacturing and seek out third-party services. So are you currently seeing this translate into increased inquiries and demands for your contract manufacturing services now that it's known that they're available?
Well, we saw it before, which was interesting to us just in terms of the investment in manufacturing. So others looking for that experience and that capability. And that was last year before kind of the acute event. I think what we're seeing now is that get expanded. So not only were they, the organizations that were looking to solve a lot of complicating problems and looking for process R&D and expanded manufacturing, I think what we're seeing now, to your point, is even more on kind of leaning into realizing that that investment we made is going to be valued to an entire ecosystem. And so we're really thinking about Wave now as how do we play a central role within the oligonucleotide ecosystem, not just on the therapeutic pipeline that we're developing, but really looking at our manufacturing capability. And more important than, because I think we're not making widgets. So I think it was too about, manufacturing, not just about equipment, but really talent, know-how, capability. And really leaning on how do we create that hub? How do we create that ecosystem that brings people in creates the opportunity to accelerate and advance meaningful medicines in new areas of biology, support that, and then increase, again, the totality of medicines around. So, I think we're seeing more and more of that. It also enables us to, by bringing in and working with others there, to continue to invest in and provide investment for that manufacturing capability so that we can sustain it. So not just coming into Wave, but really thinking about manufacturing as an investable asset that we can continue to grow.
Yeah, just to add to what Paul mentioned, I think definitely everyone is looking the speed of supplies to clinics. So that's where I think Wave has an advantage of developing these processes and provide the manufacturing opportunity for them. So.
Okay, great. Well, let me squeeze in one last question here. Paul, Shri, as your perspective as management, what do you think the market is missing most about the Wave Life Sciences story, and where's the hidden value?
Well, I think first and foremost is realization of pipeline value. Hands down. I think the data was really important. We don't minimize the impact of having target engagement and demonstrating that at low doses and seeing the durability. So I think one is just execution on delivering on the new pipeline. So starting with C9 for ALS, FTD, our HD program and our DMD program. Secondly, I think it's the advancement of our RNA editing platform led by alpha-1 antitrypsin. So not just talking about RNA editing, but delivering in terms of the first potentially therapeutic program that treats both liver, lung subcutaneously with alpha-1 antitrypsin. And then third, hence why we're discussing today, I think there's really no visibility at all to the realization that we've got an established footprint in GMP manufacturing that's ready now. So not something that can be built out for 2 years from now for validation, but a ready now facility that's poised to deliver complex oligonucleotides to patients. So when I think about the totality within Wave in terms of value recognition, it really is a 3-part story. And as we've said at the beginning of this year, those are the 3 parts we're poised to execute on in 2022.
Well, great. Thank you both for joining us today. And that brings us to the end of our session. But again, thank you for participating.
Thank you for having us.
Thank you, Kate.